Hey everyone, welcome back. In today's video, we are doing the graphical analysis of the digestive system. In this analysis, we will be considering the study of various organs of the digestive system through a graph. So on the x-axis, I am putting all the organs of the human alimentary canal, including the buccal cavity on the left. Then we have the esophagus. Then we have the stomach. Then we come across the small intestine, which includes the duodenum, which is the first part. Then we have the jejunum, and then the ileum. I'm not talking about the large intestine because there's no main digestion happening over there. On the y-axis, I have the concentration of various substances. So if you notice the blue line, that is starch. When you ingest starch, some of it is digested in the buccal cavity and converted into maltose, so maltose concentration increases. Then it stays the same in the stomach, and then gradually the concentration decreases for starch, and then maltose increases. If we look closely, what's happening here is that starch was eaten in the beginning, it was ingested in the form of bread or rice or whatever. But in the buccal cavity, there's an enzyme known as the salivary amylase, which was present in the saliva, so starch was converted into maltose. That is why maltose concentration increased in the buccal cavity. You can see the graph is a horizontal line because no digestion of starch happened in the stomach or in the esophagus because the pH was 2, which is not good for the amylase enzyme. And there is no amylase working in the stomach or the esophagus, so starch was not further digested for a while. But as soon as it entered the duodenum, you remember there was an enzyme known as the pancreatic lipase and pancreatic amylase. These enzymes helped in digesting starch further, so starch concentration broke down and it decreased. At the same time, you can see that maltose is increasing because starch makes maltose, right? So maltose and glucose increased. And then finally, the concentration was maximum for maltose and glucose. But in ileum, all the villi absorbed this maltose and glucose, so their concentration decreased again. It is because in ileum, the villi are present which absorb all the sugars into the blood flow. Now, if we consider proteins and amino acids, we are going to have a differently shaped graph. When somebody eats a diet rich in protein, proteins are not digested up until the stomach. And in stomach, they are broken down and peptides are produced. Even in duodenum, they are not digested, so the graph remains horizontal, but in jejunum, they are digested again. So let's revise what happened here. No digestion of protein happens in the buccal cavity or even in the esophagus. Why not? Because there is no protease enzyme in the buccal cavity. So no protease in the buccal cavity or obviously the esophagus. But in stomach, there's an enzyme known as pepsinogen which becomes pepsin. Pepsin is a very good protease at pH 2. So HCl and pepsin break down the proteins. So their concentration decreases and peptides are produced. That is why you can see the green line is increasing because peptides are made, and the blue line is decreasing because proteins are digested. In duodenum, you can see the concentration remained the same. Why? Because in duodenum, the pH is almost 8.5, which is not good for the protein digestion. So no digestion happens in duodenum. But in jejunum, there's an enzyme known as trypsin, which breaks down the proteins even more. So all the proteins are digested in jejunum and peptides are produced. So you can see the green line is increasing because peptides are made. At the same time, there's another enzyme in jejunum known as peptidase. Peptidase breaks down all the peptide chains to make so many amino acids. That is why you can see the highest concentration over there. But finally, in the ileum, same thing happens. All the amino acids are absorbed by the villi. So villi absorbs all these amino acids into the blood capillaries and their concentrations fall down. Moving on to lipids and fatty acids and glycerol. The orange line is lipids and the yellow line is fatty acid and glycerol. So you can see the orange line remains the same up until stomach, and then it starts to decrease only at duodenum, while fatty acid yellow line increases, and then ileum all the lines fall because lipids are not digested due to no lipase enzyme in the buccal cavity or the esophagus or the stomach. There's very minute amount of lipase over there in the buccal cavity, but it's not substantial. Only in the duodenum there's an enzyme known as pancreatic and intestinal lipase. So lipids are broken down and they are digested. While fatty acid concentration increase because obviously the lipids break down to make the fatty acids and glycerol. So fatty acid and glycerol concentration increases in duodenum and the jejunum. Bile helps in the emulsification of the lipids. And finally, all the fatty acids are absorbed in the ileum by the help of the lacteals or the lymphatic vessels in the villi. So fatty acids are absorbed in the ileum, and that is why their concentration decreases. I hope this graph made sense to you because there have been questions in the past paper where they refer to the concentrational changes. Stay tuned, guys. Thanks.